A student prepared some animal cells to view using a microscope. Figure 1 shows the student preparing the cells. So we see below it's an illustration of the student placing a cover slip. Name 2 pieces of laboratory equipment the student could have used to prepare cells to view using a microscope. And this question is worth 2 marks. So there are quite a few answers you could go for here. You could mention the microscope slide, you could mention the cover slip, you could talk about the dye or stain, and the mark scheme will also allow it if you name a specific dye or stain. You could say needle or mounted needle, pipette, scalpel, or forceps/tweezers. Figure 2 shows the student's light microscope. So here we have figure 2, we have 2 labels, part A at the top and part B lower down. And the question says, name part A, and is worth 1 mark. So part A is the bit that you look down when you're using the microscope, and this can be called the eyepiece or the lens. What is the function of part B? And this question is worth 1 mark. So part B can be twisted and it adjusts the position of the stage. And the reason for this is that it allows you to focus the image. The student tried to look at the cells using the microscope. Suggest 1 reason why the student could not see any cells when looking through part A, and this is worth 1 mark. So most of the potential answers for this question will be to do with how different parts of the microscope are positioned. It could be that there are no cells in the field of view, so that's to do with where the student has focused. It could be that the slide is not in the right position, the mirror on the microscope itself might not be in the correct position, or the objective lens may be dirty or not clicked into the correct position. It could be that the student is looking at an air bubble, so there's just a bit of a gap in the specimen itself. Or it could just be that the microscope is not in focus. Red blood cells are specialised animal cells. Compare the structure of a red blood cell with the structure of a plant cell, and this question is worth 6 marks. To get full marks on this question, it's not just about listing out 6 points. You need to name the scientifically relevant features, and you also have to show in your answer the magnitude of similarity. So if we start with the differences, we know that a red blood cell, and we can abbreviate that to RBC, has no nucleus, whereas a plant cell does, and we can abbreviate plant cells to PC. We can say that the red blood cell has no cell wall, and this is because it's an animal cell, whereas the plant cell does. Healthy red blood cells are consistently in a biconcave shape, so that means it's kind of like a donut but without a hole in the middle, just an indentation. Plant cells, on the other hand, come in all sorts of different shapes and sizes according to their function. And because this is a structure question, it can be really helpful to just remember your basic diagrams of a plant cell and a red blood cell. It might also be helpful to just remember the functions of these particular cells. The functions aren't needed to get yourself the marks, however they're very closely tied with the structures of the cells, so it might help jog your memory. For example, the red blood cell has no nucleus, so it can contain more hemoglobin, it has no cell wall because it doesn't need that support, and it's biconcave for greater surface area and so it can fold and fit through narrow capillaries. But when you're writing, just remember to keep your focus on those structural features. More differences that you could mention are that the red blood cell contains hemoglobin, and this helps it to carry oxygen. The plant cell does not need to do this, so it doesn't need the hemoglobin. Similarly, the plant cell contains chlorophyll, and this is because it's got to photosynthesize. The red blood cell is an animal cell, so it does not need to do this. For the same reason, red blood cells don't contain any chloroplasts, so these are chlorophyll-containing organelles. It's also only plant cells that contain a permanent vacuole, so these are single-membraned organelles and they help the plant cells with storage and also help them maintain rigidity. Lastly, you can make a comment on the overall size difference, so red blood cells are much smaller than plant cells. And note that all of the points we've made so far have stayed irrelevant to the question because we're just talking about the structural features. Now we can move on to talking about some of the similarities. Both the red blood cell and the plant cell have a cytoplasm, they both have a cell membrane, and both of them contain pigments, although these are different types of pigment. So now we have plenty of statements, and we can check against those mark scheme specification points. So we've got scientifically relevant features, and we've also got a magnitude of similarity. We can see that when we say both contain pigments, although they are different, and you can compare that against the times where we've got absolute similarities. Both have cytoplasm, or one has no cell wall whereas the other does. We have comparisons of physical shapes, and we also have comparisons of the contents of these cells. And you can see we have well over 6 points. You don't need every single one of these to get the marks. The only other thing worth noting is that you do need to have a balanced argument. You will not be able to just list 6 differences and get all of 6 marks. You need to have some from the differences and some from the similarities. When placed into a beaker of water, a red blood cell bursts and a plant cell does not burst. Explain why the red blood cell bursts but the plant cell does not burst. And this question is worth 2 marks. The answer the examiner wants from you here is that water enters the cells by osmosis. And remember, osmosis is the movement of water across a partially permeable membrane from an area of low solute concentration to an area of higher solute concentration. Or you could say from a dilute solution to a concentrated solution. The mark scheme will allow you to say diffusion here, however just bear in mind that this does not specifically mean the movement of water. The second mark you get by stating that the plant cell has a cell wall, and this is what prevents it from bursting. A student investigated the effectiveness of 3 different antibiotics. Figure 3 shows how the student set up an agar plate. So below is figure 3, we can see we've got disc A, which has antibiotic A, disc B, which has antibiotic B, disc C that has antibiotic C. The pale grey shading is the bacteria growing on the agar, and the largest circle is the petri dish. The student used aseptic techniques to make sure that only one type of bacterium was growing on the agar. Describe 2 aseptic techniques the student should have used, and this question is worth 2 marks. So here we're being asked about our experimental technique, and first of all let's double check our definition of an aseptic technique. These are precautions that you take while carrying out your experiment to make sure that the microorganisms you're investigating do not escape the vessel or become contaminated by other microorganisms. Ways you can go about this include sterilising all of your equipment and all of your surfaces. So if you're using an inoculating loop, you might sterilise it using the flame of your Bunsen burner, and you can spray your surfaces with a disinfectant. You might use sterilised agar in your agar plate. You can secure the lid of your petri dish with tape, so this stops other microorganisms getting in. However, you need to make sure you do not completely seal the petri dish because oxygen does need to be there for the organisms inside to be able to breathe and grow. Lastly, when they are placing their discs A, B and C, they make sure to only lift the lid just a little bit so that nothing else can fall in from the outside air. The student placed an agar plate in an incubator at 25 degrees Celsius for 48 hours. Figure 4 shows the agar plate after 48 hours. So here we have figure 4, we've got our overall petri dish and we've got the bacteria growing on agar still represented by the pale grey. Then the white zones represent the areas where no bacteria is growing. Which antibiotic is the least effective? Give a reason for your answer, and this question is worth 1 mark. So to determine which is the least effective, we have to look at these white zones around each disc, and these zones are called the zones of inhibition. The larger the zone of inhibition, the more bacteria the antibiotic has killed, and therefore the more effective it is. When we look at figure 4, we can see that those white rings around each disc are of different sizes. B is the smallest, then A, and then C is the largest. The question asks us for the least effective antibiotic, and that means we need to look for the smallest zone of inhibition, which we can see is around the disc with antibiotic B. The reason you need to give is that B kills the fewest bacteria, or it has the smallest area where there are no bacteria growing. Calculate the area where no bacteria were growing for antibiotic C. Use pi equals 3.14 and give the unit. This question is worth 5 marks. To answer this question, you need to measure the diameter across the disc that has antibiotic C and the zone of inhibition around it. Then you have to remember your formula for the area of a circle, so that's pi r squared, and in this case we use d over 2, so we just halve the diameter to give us the radius. So you go back to figure 4, measure the cross-section across antibiotic C, and that will be printed to a specific size on your paper, and you'll get a diameter of 2.2 centimetres. We can now go back and plug that into our equation of pi d over 2 squared. So 2.2 divided by 2 will give you the radius. It doesn't matter if you do that ahead of plugging it into the equation or if you just substitute it straight in. But just make sure if you leave it as 2.2 over 2, you put it in brackets before you square. Plug that into your calculator and you get an area value of 3.799 and so on. But we're not done yet. We can round that to a sensible value and make sure we remember the unit. The question specifically asked for that. Earlier we took our diameter measurement in centimetres, and therefore our radius measurement is in centimetres, and then we square that because of our equation, which means our final unit is centimetres squared. If we give this to 3 significant figures, we have to do a little bit of rounding. We get 3.80 centimetres squared, and that is your final answer there. With 1 mark for a correct diameter or radius measurement, another for remembering your formula for a circle and correctly plugging it in, and the last 2 for your numerical answer and the unit. Suggest one way the student could improve the investigation, and this is worth 1 mark. So you need to come up with some ways that they could get a better result. So they can carry out repeats and calculate the mean. That increases the likelihood of an accurate result. Similarly, they could carry out repeats and eliminate the anomalies, so an anomaly is an outlier that doesn't fit with the pattern. They could use a control disc, so this is one that's got no antibiotic on it, or they could try again with different types of bacteria. Body mass index, or BMI, is a way of finding out if a person's body mass falls within a healthy range for their height. Table 1 shows information about 2 people. So below is table 1, we have person A and person B. Then we have each of their body masses and each of their heights. And then that is used to calculate BMI in the final column. Figure 5 shows 5 BMI categories for adults. So this is figure 5. On the y-axis we have height in metres, and on the x-axis we have body mass in kilograms. And each of the different colours of shading represents a different category. There's underweight, normal, overweight, obese, and clinically obese. Which is the BMI category of person A in table 1? Tick 1 box, and this question is worth 1 mark. So we refer back to table 1, we're looking at person A, so the values are 63 and 1.65. Now we need to plot these on the graph and figure out which category the point lies in. It turns out to be in the light grey area for normal, and so that is our answer. Calculate value x in table 1. Use the equation BMI equals body mass over height squared. Give your answer to 3 significant figures, and this question is worth 3 marks. First, let's return to the table to get our data to calculate x. Remember, BMI is equal to body mass over height squared. it's important you don't plug them into the equation the wrong way round. So the values we want are 92 and 1.71, and that's the value that we square. We then put these into the equation, and we get an answer of 31.46 and so on. But we're not done yet. The question specifically says to give it to 3 significant figures. So that means our final answer will be 31.5, remembering to round the 6 up. Here you get 1 mark for plugging the correct numbers into the equation, another for that 31.46 answer, and another for your final answer to 3 significant figures. Scientists think that there is a link between BMI and life expectancy. Table 2 shows information about predicted life expectancy of men after the age of 50. So below is Table 2, we have the BMI categories like what we saw in the graph. Then in the first column is the predicted number of years living in good health after 50, and the second column is the predicted number of years living in bad health after 50. Describe 2 patterns shown in Table 2 about the effects of BMI category, and this is worth 2 marks. So this question is giving you 1 mark per reason. You don't need to explain anything, you just say what you see. So the first option, you could put the higher the BMI, the lower the number of years living in good health. You can see the person in the normal BMI category lives for 19.06 years, whereas the person who is clinically obese lives for 13.07. Alternately, you can look at the second column. The person in the normal category lives 4.98 years in bad health, whereas the clinically obese person spends 10.10 years in bad health. So you can say the higher the BMI, the higher the number of years living in bad health. The mark scheme will also allow a comment about the total life expectancy, which you get by adding up each row. So the higher the BMI, the lower the total life expectancy. The number of people who are obese in the UK is increasing. Explain the financial impact on the UK economy of an increasing number of people who are obese, and this question is worth 2 marks. What the examiner is looking for you to mention here is the cost to the NHS, and the terminology you can use is quite varied. You could say NHS, health service, government or hospitals, but the point you need to get at is that these services have to pay out more because obesity leads to a variety of health problems, and such health problems may need additional surgeries, medication, or prolonged hospital stays in order to treat them. You can be credited for mentioning secondary conditions such as strokes or diabetes. Alternatively, you could talk about the effects of people with obesity being unable to work due to health problems. If they're unable to work, then they're not earning, and so the employer or the government has to provide financial support. A person who is obese is more at risk of arthritis. Arthritis is a condition that damages joints. Suggest how arthritis could affect a person's lifestyle, and this question is worth 1 mark. Arthritis can be very painful, and because it affects the joints, many people who suffer from it can experience movement issues. They may be unable to carry on working and thus unable to carry on earning money, and there can also be a mental health impact, whether this is from their inability to move around and continue to exercise a lot, or from the pain of the condition. Lastly, they may need to frequently visit the doctor, take medications, or have surgeries done. A person who eats a diet high in saturated fat may become obese. Name 2 health conditions that might develop if a person eats a diet high in saturated fat. Do not refer to arthritis in your answer, and this question is worth 2 marks. The first thing you can talk about here are heart diseases. So this might be cardiovascular disease, which you can write as CVD, or coronary heart disease, which is written as CHD. The mark scheme will accept it if you just put heart disease or heart attack or a stroke. The second option you can put is type 2 diabetes, Aside from these main options, the mark scheme also allows cancer, liver disease, or blood pressure/blood cholesterol being too high. All living organisms respire. What is the chemical equation for aerobic respiration? Tick one box. And this question is worth 1 mark. First, let's check our key term aerobic definition. So this is respiring with oxygen. Anaerobic respiration is respiring without oxygen. We can use this information to start eliminating any of the options that do not have oxygen in its form O2. Next, we just work our way down the list. So the first option, we can see that oxygen and carbon dioxide are both on the same side of the equation. And this isn't possible in respiration, seeing as we inhale oxygen and exhale carbon dioxide, so that can't be correct. The next option we'd already eliminated because it doesn't have oxygen in the form O2. The third option says water plus carbon dioxide gives oxygen and glucose. This is the reverse of respiration, it's photosynthesis. This means by process of elimination, the last option must be correct. And you can double check it by looking at the balancing. So on either side there are 18 oxygens in total, there are 6 carbons in total, and there are 12 hydrogens in total. Name the subcellular structures where aerobic respiration takes place. This question is worth 1 mark. And the answer is in the mitochondria. Energy is released in respiration. Give 2 uses of the energy released in respiration, and this question is worth 2 marks. There are a few potential answers you could have here. One is muscle movement or contraction, another is keeping warm, another is active transport, which is light diffusion, only moves against the concentration gradient, which is why it requires energy. And the last could be building up larger molecules in the body. Describe 2 differences between aerobic and anaerobic respiration in humans. Do not refer to oxygen in your answer, and this question is worth 2 marks. So bear in mind when answering this question that we're talking about animals, we're talking about humans. And anaerobic respiration in humans will produce lactic acid, whereas aerobic respiration will not produce lactic acid. And it's important here that you use that comparison word whereas. Another difference you could point out is that aerobic respiration produces carbon dioxide in animals, whereas anaerobic does not produce carbon dioxide. Aerobic also produces water, whereas anaerobic does not. Lastly, you might talk about the energy output of both. Anaerobic respiration releases less energy than aerobic respiration, and the comparative word here is less, so make sure you include that. What are the 2 products of anaerobic respiration in plant cells? Tick 2 boxes, and this question is worth 2 marks. Remember, anaerobic respiration is when you are not taking in oxygen. Working down the list, the first option is carbon dioxide, and in plants this is correct. The second option down the list is ethanol, and this is also correct. Carbon dioxide and ethanol are the products of fermentation, the process that occurs when plant cells respire anaerobically. To eliminate the others, you can look at glucose and know that that is not a product of respiration, that is a reactant. Lactic acid is produced in animal cells, not plant cells, and water is only a product of aerobic respiration. So carbon dioxide and ethanol are your final answers. A scientist investigated respiration and photosynthesis using some pondweed and a pond snail. Figure 6 shows the apparatus used. So below is figure 6 and labelled is a data logger, a carbon dioxide probe, some pondweed, a pond snail, and some pond water in a boiling tube. The apparatus was left in a well-lit room for 5 days. The data logger recorded the concentration of carbon dioxide continuously. After 5 days, the scientists completely covered the boiling tube with black paper. The data logger continued to record the concentration of carbon dioxide. Figure 7 shows the concentration of carbon dioxide inside the boiling tube over 15 days. Below is Figure 7, the y-axis shows concentration of CO2 and the x-axis shows time in days. Explain why the concentration of carbon dioxide in the tube stayed the same between day 0 and day 5, and this question is worth 2 marks. So the reason the concentration of CO2 is remaining constant is because it's being produced and consumed at the same rate, and you have to explain how this is happening. So the pondweed takes in CO2 for photosynthesis, but the snail and the pondweed also respire, producing CO2, so it balances out. Suggest why the concentration of carbon dioxide increased between day 5 and day 10, and this is worth 1 mark. We read that day 5 in this experiment is when the test tube was covered by black paper, so no light was reaching the organisms inside. No light means no photosynthesis, so the plant would not be taking in carbon dioxide. However, the snail and the plant are still respiring, meaning they're still producing CO2 and causing an overall increase. On day 10, the pond snail died. Explain why the death of the pond snail caused the concentration of carbon dioxide to increase after day 10, and this question is worth 3 marks. So the connection the examiner is looking for you to be able to make here is to microorganisms and decomposers. The snail has died and is therefore attracting bacteria and decomposers. These then break down the snail, and while they do so, they respire. And when they respire, they release CO2, which is the reason for the increase after day 10. So you get 1 mark for saying the snail is being decomposed, another for saying this is done by decomposers or bacteria, and the last for saying that their respiration releases CO2. Amylase is an enzyme that breaks down starch. Amylase is a polymer of smaller molecules. Name the type of smaller molecule, and this question is worth 1 mark. So it's told us that amylase is an enzyme, and that means that it is a protein, and proteins are made up of the monomers amino acids, so that's the answer you want to put here. Name 3 parts of the human digestive system that produce amylase, and this is worth 2 marks. The answers you can put here include the salivary gland, the pancreas, and the small intestine, and make sure you specify this is the small intestine, not the large or just the intestine. Explain how amylase breaks down starch. Answer in terms of the lock and key theory, and this question is worth 3 marks. So what happens here is the starch, which is the substrate, binds to the active site of the enzyme, which is amylase. So enzymes have a specifically shaped region, and this is what is meant by the active site. In turn, the substrate also has a specifically shaped binding site, and it is complementary to the active site of the enzyme, so it fits together kind of like 2 puzzle pieces. And this is what you need to explain when you're talking about the lock and key theory. So the shape of the enzyme active site and the substrate are complementary, that means they fit together. And what happens is a chemical reaction occurs to produce smaller molecules. Remember, an enzyme is a biological catalyst, so its entire function is to speed up reactions like this one. The mark scheme also allows an alternative way of describing that last point. You can say that the bonds between the starch molecules are broken, and this is what produces those smaller monomers. So you get 1 of your marks for talking about the active site, another for saying that it's complementary to the substrate, and the final one for describing the chemical reaction that produces the smaller molecules. And that could be just by stating that there are bonds broken. A student investigated the effect of temperature on the activity of amylase. Figure 8 shows the apparatus used. So here we have figure 8, and we have test tubes sitting inside a beaker of water at 5 degrees C. In one test tube is 5 centimetres cubed of starch solution, and in the other is a centimetre cubed of amylase solution. This is the method used. Set up the apparatus as shown in figure 8. After 5 minutes, pour the starch solution into the amylase solution and mix. Remove 1 drop of the starch amylase mixture and place onto a spotting tile. Immediately add 2 drops of iodine solution to the starch amylase mixture on the spotting tile. Then record the colour of the iodine solution added to the starch amylase mixture. Repeat steps 3 to 5 every minute until the iodine solution stays yellow-brown. Repeat steps 1 to 6 using water at different temperatures. Name 2 control variables the student used in the investigation, and this question is worth 2 marks. Remember, control variables are kept the same, and one of these you could talk about is the time before mixing solutions. This is called equilibration time. Another control variable was the volume of starch solution, or you could talk about volume of amylase solution, mixture added to the spotting tile, or the volume of iodine added. Why did the student leave the starch solution and the amylase solution for 5 minutes before mixing them? And this question is worth 1 mark. So what it's asking you about here is that equilibration we mentioned earlier, and it's essentially to allow the solutions to reach the same temperature. So that will be the temperature of the water. And you can say it like that, or you can say to allow both solutions to reach 5 degrees C. Table 3 shows the results of the investigation. So here is table 3, the left-hand column has the temperature in degrees Celsius, and the right-hand column is the time taken until the iodine solution stays yellow-brown in minutes. What conclusion can be made about the effect of temperature on amylase activity between 20 degrees C and 65 degrees C? And this is worth 1 mark. Alright, so we have to look at this region right here and see if there is a trend. Between 20 and 35 degrees, the time taken decreases, which means the activity increases, the reaction happens faster. Then between 35 and 50, we get an increase, so the reaction starts to slow down. And this continues to happen between 50 and 65 degrees. So we can state that as temperature increases, enzyme activity also increases up to 35 degrees Celsius. Then after that point, activity starts to decrease, and we don't need to explain why this happens, we're just stating the effect. Explain the results at 5 degrees C and at 80 degrees C. Use table 3, and this question is worth 5 marks. So we have a copy of table 3 on the left, and we can see that at 5 degrees C and at 80 degrees C, the solution did not become yellow-brown at all. And the bit of information that you need to be able to talk about this is the fact that iodine will turn a dark blue-black in the presence of starch, but it will remain yellow-brown if there is no starch. So your first statement could be that iodine is not yellow-brown, it would still be that dark blue-black colour, because the starch is still present. It has not been broken down by the enzyme amylase. Next, you need to start explaining why this has happened. So 5 degrees C is a very low temperature, and the molecules of the amylase or the molecules of starch would have a low kinetic energy. And this means that the frequency of their collisions will decrease. They bump into each other fewer times per second, and the reaction therefore occurs more slowly. 80 degrees C, on the other hand, is a very high temperature, and at this temperature the amylase has been denatured. So ordinarily amylase will have its specifically shaped active site, and this is complementary to the substrate. However, if you go too far over its optimum temperature, the active site will denature, and this means it will no longer be the shape that it's supposed to be, and is therefore no longer complementary to the substrate. So in your answer, you just have to make a statement that the substrate does no longer fit. Overall, you get your first mark for saying that the starch is still present. The second is talking about the low kinetic energy at 5 degrees. The third is about enzyme-substrate collisions. The fourth is about denaturation, and the fifth is the consequence of that denaturation. The student investigated the effect of temperature on amylase activity. Describe how the student could extend the investigation to determine the effect of a different factor on amylase activity, and this is worth 2 marks. Well, first of all, they have to keep the temperature constant. This is now a control variable, so it can't affect the investigation. But they have to change something different, and this could be the enzyme concentration or the substrate concentration. Figure 9 shows a cross-section of a leaf. Below is figure 9, and we can see we have different cells labelled A through to D. Which cell is the most transparent? Tick 1 box, and this question is worth 1 mark. Well, the answer to this is A, because A is the topmost cell and the light must pass through it in order to get to other photosynthesizing cells. Which cell structure in a leaf mesophyll cell is not found in a root hair cell? And this question is worth 1 mark. The answer is the chloroplasts, because chloroplasts would have no use in a root hair cell as they get no sunlight, they're underground. Plants lose water through their leaves. Name the cells in the leaf that control the rate of water loss, and this question is worth 1 mark. The answer is the guard cells, and these lie on either side of the stomata and control how widely or frequently it opens. Water is taken in by the roots, transported up the plant and lost from the leaves. Which scientific term describes this movement of water? And this question is worth 1 mark. The answer to this is the transpiration stream. Which change would decrease the rate of water loss from the plant's leaves? Tick 1 box, and this question is worth 1 mark. When you're answering this, it can be helpful to think about evaporation, which is a way that water can be lost, and think about the factors that affect it. So you can have the difference in humidity, the temperature, and the airflow surrounding the area. Remember, we're looking for the options that will decrease the rate of water loss. If we look at the first one, increased humidity, that will mean that there is less of a difference between our area where there is water and the surrounding environment. Humidity means there's lots of water in the air, and so we know that that will decrease the rate of water loss. The next one, increased light intensity, well, that will increase water loss. Increased density of stomata, so those are the holes in the underside of the leaf, they will also increase water loss. And finally, the last one, increased temperature, well, that will also increase the rate of water loss. So we know that the top one is the only correct option. Compare the structure and function of xylem tissue and phloem tissue, and this question is worth 6 marks. Remember that in order to get full marks on a question like this, you can't just list 6 statements. You have to identify scientifically relevant features, and then you have to state how they are similar or different, and then you also have to state the magnitude of similarity or difference, whether it is absolute or only in certain conditions. You might start by talking about the structure of these 2 types of cell. Xylem is made up from dead cells, while phloem is made of living cells. Phloem cells have pores in their end walls, whereas xylem do not have pores in the end walls. Xylem is hollow, while phloem contains cytoplasm, and xylem contains lignin, but phloem does not contain lignin. And notice how when we're describing the differences here, we say one has X, whereas the other has Y. Make sure you include those connectives. Now, moving on to the similarities in structure, you could say that both are tubular and both are made of cells. Moving on, we can talk about some of the functional aspects. The xylem transport mineral ions and phloem transport dissolved sugar. Xylem is involved in transpiration, while phloem is involved in translocation. Xylem transports unidirectionally, that means in only one direction, while phloem transports bidirectionally, that's in two directions. In terms of functional similarity, you could say that both transport liquids or substances along the stem, leaves, roots, or just the plant. Make sure that you're talking about both structure and function in your answer, and make sure that you have enough scientifically relevant features to make it worth 6 marks. We've stated how they are different or similar, and we've also stated the magnitude of similarity or difference. Lastly, make sure you include similarities and differences in both your structure paragraph and your function paragraph. Figure 10 shows the total volume of water lost from a plant over 6 hours. So below is figure 10, the y-axis shows the volume of water lost, and the x-axis shows the time of day. It also shows that a tangent has been drawn to a certain point on the line of the results. Determine the rate of water loss at 12 o'clock. And you can see on the graph on the left that 12 o'clock is the point where the tangent has been drawn, and the question tells you to use that tangent and to give your answer in centimetres cubed per minute and in standard form. And this question is worth 4 marks. To determine the rate of the water loss, you will need to use the following formula, which is change in y over change in x, and you apply this to the tangent. You want to take points from as far apart on the tangent as possible, so we can pick the top right, where the volume is 230 centimetres cubed. Then we look at the bottom left, which is 190 centimetres cubed, and the difference between these two numbers is our change in y. Now we get our x values, and make sure you put them down in the same order you put down the y values. You should also remember that the question wants you to answer in terms of minutes, and you can make this conversion either now or later on in your working. The time of 3 o'clock corresponds with the value of 230, so we write that down first, and then the time of 8 o'clock corresponds with the value of 190. Now you might decide to simplify your equation and do your conversions. So 7 hours is the difference between 3 o'clock and 8 o'clock, 7 times 60 is 420 minutes, and the difference between 230 and 190 is 40. So the rate of change is 40 divided by 420. That gives you an answer of 0.09523 and so on, and this is in centimetres cubed per minute. But we're not done yet, we still have to put it in standard form. And standard form looks like this, it's x times 10 to the power of y, when x is between 1 and 10. So in this instance, we can write out our number 0.09523, and that becomes 9.523 times 10 to the power of minus 2. And we know it's to the power of minus 2 because that decimal point has moved 2 places to the right. So you get one of your 4 marks for that rate of change equation, another for getting an answer to that that's correct, the next one for carrying out your conversion and making sure that it's in minutes, and the last for correctly putting your answer in standard form. The rate of water loss at midnight was much lower than at 12 o'clock. Explain why, and this question is worth 2 marks. The answer is that at midnight the stomata are almost completely closed because it is much cooler or because there is less light. Figure 11 shows where 3 of the same type of tumour were found in a patient. So below is a diagram, it's figure 11, and it shows dark patches representing tumours. Malignant tumours are cancers. Describe what happens to cells when a tumour forms, and this question is worth 1 mark. The answer is that cells grow or divide abnormally or uncontrollably. What evidence is there in Figure 11 to suggest that the tumour in the lung is malignant? And this question is worth 1 mark. First of all, you can identify the lungs on the diagram and see that the largest grey patch representing a tumour is in one of the lungs. However, we can also see 2 more grey patches, and these reside in the liver. So we can say that the tumour is malignant because it is seen to have spread to other organs. A person with one of these types of cancer may experience symptoms such as tiredness, frequent infections, and bleeding that will not stop after the skin is cut. Explain how a very low number of blood components in the body can cause these symptoms. And the question is worth 6 marks. Now, to get full marks here, you can't just list 6 points. You must identify relevant points, you must add a suitable amount of detail, and they must be logically linked. Starting with the symptom of tiredness, you can say that fewer red blood cells means that less hemoglobin will be transported around the body. Hemoglobin is a carrier of oxygen, so that means that oxygen will not be transported around the body either. Less oxygen means less aerobic respiration and therefore less energy released. Furthermore, lactic acid will be produced, and this causes muscle fatigue. Moving on to talk about the next symptom, frequent infections. This is because fewer white blood cells are in your system, and the mark scheme will allow you to call these white blood cells phagocytes or lymphocytes as well. Fewer white blood cells in the system mean that fewer antibodies are being produced, and less phagocytosis, that's the engulfing of a pathogen by a white blood cell, is taking place. Fewer pathogens are therefore killed. The last symptom that it wants you to talk about is excessive bleeding, and this is caused by a lack of platelets in the bloodstream. So platelets are cell fragments and they're quite sticky, which allows them to clump together and block any holes that the blood is leaking through, so this process is called clotting. So overall, you don't have to mention every single one of these points to get your full 6 marks. You can see we've got well more than 6 there, but they do have to link together logically, and you need to have enough detail that it all makes sense and is relevant to the question. Just make sure that you mention all of the symptoms. If you only mention tiredness and infection, say, you won't get full marks. Some patients with a very low number of blood cells may be given a blood transfusion. A blood transfusion is where a patient receives blood from a donor. Different people have different blood groups. Figure 12 shows the red blood cells found in people with different blood groups and the antibodies that can be made by people with different blood groups. So having a look at figure 12, we can see it's a kind of chart, and we can see that blood group A has red blood cells with type A antigens, and it has anti-B antibodies. Blood group B has type B antigens on the red blood cell and has anti-A antibodies. Then blood group O doesn't have any antigens, and it has anti-A and anti-B antibodies. Antibodies can bind to antigens that have complementary shapes. When antibodies bind to the antigens on red blood cells, many red blood cells begin to clump together. Each red blood cell is about 8 micrometers in diameter. Many capillaries have an internal diameter of about 10 micrometers. In one type of blood transfusion, only red blood cells from a donor are transferred to the patient. It is dangerous for a patient with blood group A to receive red blood cells from a donor with blood group B. Explain why, and this question is worth 3 marks. So the point the examiner wants you to get at is anti-B antibodies will bind to type B antigens on the donor's red blood cells. So if we have a look back at the diagram, we can see that there are anti-B antibodies present in the blood of the donor with blood group A, and these anti-B antibodies have that square shape, which is complementary to the type B antigens present in the person with blood group B. So what happens is the red blood cells start to clump together, and this causes them to be wider than the capillaries and start to block them. The result of this is oncoming red blood cells cannot deliver oxygen or glucose to other cells of the body, and this means that those cells cannot respire. And that's all you need to say, it's a mark per statement. Explain why blood group O red blood cells can be given to patients with any blood group, and this question is worth 2 marks. To go about answering this, you might want to have a look back at the chart from earlier. If you look at the red blood cells for the person with blood group B, for example, you can see that there are type B antigens on the surface of the red blood cell. But if you look at the red blood cells of somebody with blood group O, you can see that there are no antigens on the surface. And this means that the antibodies present in the blood of a person with blood group A or blood group B cannot bind to the blood group O person's red blood cells. The way you describe this in your answer is by saying that no antigens present on the type O red blood cell means that antibodies cannot bind to it. Table 4 shows some of the risks associated with blood transfusions. So below is Table 4, the left-hand column shows risks, and the right-hand column shows the probability of that risk occurring. Which risk has the lowest probability of occurring? Tick one box, and this question is worth 1 mark. So what you'll want to do is have a look back at the table and convert all of those probability values into a form where they're easy to compare. So we can turn them all into percentages, for example. 0.9 is already a percentage. The next one you do 1 over 3 times 10 to the power of 5, and then you multiply that by 100, and that will give you 3.33 times 10 to the minus 4%. The next one you just times that by 100, and that gives you 6.7 times 10 to the minus 5%. And the next one is 1 over 70,000 times 100, and that's 1.428 times 10 to the minus 3%. So the smallest one is the one that's to the power of minus 5. The hepatitis C infection. A person has a tumour blocking the tube leading from the gallbladder to the small intestine. Explain why this person would have difficulty digesting fat, and this question is worth 5 marks. So what's happened is this blockage is causing no or less bile to reach the small intestine. So there is less emulsification of fat, and it isn't beading into little droplets. This reduces the surface area to volume ratio, and means that lipase, an enzyme, cannot break down the fat so efficiently. The pH of the small intestine is not neutralised or remains alkaline, meaning lipase is not operating at its optimum pH, so that's another reason it can't break down the fat so efficiently. And now you can see we've got 5 statements written down and you get 1 mark for each.